The description of the double helical structure of DNA deoxyribonucleic acid by Whitson and Crick in 1953, The Facts of Life. West Islandmer in Science History. Their discovery sparked of a new era in scientific research, which has had and will continue to have far-reaching consequences. A polymer of nucleotides. Each DNA star is a polymer made up of nucleotide subunits. The nucleotides join together to form long underbranch polynucleotide chains. Each nucleotide consists of deoxyribose, a 5-carbon or pentose sugar, and organic nitrogen-containing base, of which there are four different types, and phosphoric acid. The sugar and the organic base join together by a condensation reaction to form a nucleotide, and a condensation reaction results in the removal of water molecule. Another condensation reaction joins to the nucleotide with phosphoric acid to form the nucleotide. This bond from between carbon 5 of the sugar and the phosphate and is such a phosphor bond. The organic bases present in DNA are parents, guanine G and adenine A, or pyrimidines cytosine C and thymine T. Purines have a double ring structure. Pyrimidines have a single ring structure. Two nucleotides can join together by a condensation reaction between the phosphate group of one nucleotide and the hydroxyl group on carbon 3 of the sugar of the other nucleotide. The bonds linking the nucleotides together are strong, covalent phosphodiester bonds. The process can be repeated so that a polynucleotide can bulge up. The cane has a sugar phosphate backbone with the organic bases projecting outwards. Its cane has two discant ends, a 3 prime and a 5 prime, and at the 3 end the carbon 3 of the deoxyribose is closest to the end. At the 5 end the carbon 5 of the deoxyribose is closest to the end.
The double helix DNA consists of two polynucleotide chains coated around each other to form a double helix. The double helix is held together by hydrogen bonds between parts of bases in the two chains. Depends on the species of the bases. A pairing can only bond with a pyrimidine and on their ability to form hydrogen bonds. Adenine, a pairing, pairs with thymine, a pyrimidine, forming two hydrogen bonds A equals T. Guanine, a pairing, pairs with cytosine, a pyrimidine, forming three hydrogen bonds G equals C. Complementary base pairing. These complementary base pairs are the only ways the bases can bond and join the two nucleotide chains. Thus, the sequence of bases along one polynucleotide chain determines the sequence along the other. An adenine one chain means there must be a thymine on the other can and the point, and so on. Complementary base pairing from the basis of DNA replication and its ability to form messenger RNA during protein synthesis. Complementary base pairing can happen only if the two polynucleotide chains are antiparallel. Antiparallel chains run in opposite direction. One chain runs from 3 to 5 and the other 5 to 3. Watson and Crick's model of DNA showed that the base pairs are 0.34 nanometers apart, and that each complete turn of the helix has 10 base pairs. In summary, DNA is a double helix made of two polynucleotide chains. Each chain has a sugar phosphate backbone on the outside with organic bases on the inside. The two chains are held together by complementary base pairing. The chains are antiparallel. The 5 end of one chain lies next to the 3 end of the other chain.
What is in a chromosome? A chromosome consists of hundreds or thousands of genes. A gene is the basic unit of inheritance, and specialized parts that are thought to be important to the chromosome's stability and function. The deoxyribonucleic acid DNA that makes up the genes is packaged with the aid of proteins to form a complex structure. Chromosomes also contain small amounts of ribonucleic acid, RNA. DNA is packaged in chromosomes. Each human chromosome contains one very long DNA molecule which unraveled will measure about 4 and 8 cm in length. The total length of DNA in the nucleus of a human cell has been estimated to be about 2 and 2 m. This poses a packaging problem. How does a chromosome measuring on average 6 pm long contain about 8,000 times its length of DNA? The answer is that chromosomal DNA is intrinsically folded and is tightly bound to protein molecules and called histones. Histones are small proteins that are rich in the amino acid lysine and or arginine. The complex formed between DNA and histones is called chromatin. Chromatin takes up stain and is visible in non-dividing nuclei. Individual chromosomes can be seen under the light microscope only during cell division, mitosis or meiosis. Nucleosomes. Each DNA molecule is wound around histones arranged in groups of 8 known as octamers. The DNA in octamers form bead-like structure known as nucleosomes. Positively charged groups on the side chains of the histones form strong ionic bonds with negatively charged phosphate groups in the backbone of DNA. In each nucleosomes, a length of DNA containing about 115 base pairs is wrapped around the octamer. Another histone molecule attached to the outside of the nucleosome binds DNA to the octamer. The nucleosome is regarded as the basic unit of the structure. The linker region, the thread of DNA between the nucleosomes, varies in length from 14 to over 100 base pairs.
Nucleosomes fold to form solenoid fibers. More histones in the linker region help to fold the thread of DNA and nucleosome, the nucleosome fiber, into a tightly structure called a solenoid. The solenoid are thought to be further looped and coiled around non-histone protein called scaffolding proteins. The precise details of this higher level of folding are not known. The centromer. Each chromosome has a centromer which usually appears as a constriction when the chromosome condenses during mitosis and meiosis. The position of the centromer can be used to distinguish between different chromosomes. Centromeres do not contain any genes. However, they do contain large segments of highly repetitive DNA, called alpha satellite DNA. This is thought to play a significant role in centromere function. The centromere contains the kinetochore. This is densely tiny structure that attaches the chromosome to the spindle apparatus during nuclear division. Centromeres control the distribution of chromosome during cell division. Chromosomes that do not have centromeres cannot divide. Telomeres are located at the ends of chromosomes. They consist of DNA and protein. The telomeres appear to play a vital role in maintaining the stability of the chromosome, sealing the ends of linear DNA. They have been likened to the tips of shoelaces and have a similar function. To stop the DNA fraying, they also seem to play an important role in regulation cell division. Under normal circumstances, telomeres become shorter and shorter with each cell division. When the telomeres have shortened to a certain critical length, the cells stop dividing. If the telomeres are removed, the chromosome disintegrates. It is thought that the aging process may be linked to telomere damage. Telomeres are all in sensor. Telomeres contain repeating sequences of bases which are synthesized with the help of an RNA containing enzyme called telomeres. Telomeres activity is suppressed in normal human somatic body cell. However, in sensory cell, telomeres is active and maintains the telomere length so that the cell continues to divide. It is thought that this abnormal retention of the telomeres is involved in the development of some types of cancer. Thank you.